Today we learn about gastrointestinal hormones. Our body have different systems, and the function of all these systems needs to be controlled, regulated. So all these body functions are regulated, controlled by two mechanisms. One is neural mechanism, another is humoral mechanism. Neural mechanism is executed by nervous system, and the hormonal control is done by endocrine system. The control of nervous system on functioning of different organs are very fast, rapid, but short acting. But for humoral control, means endocrine control, initiation takes time. Control initiation takes time. Mechanism is little bit delayed, and their effect will be lasting longer than the neural control. So these are the two basic differences between the two mechanisms. Digestive system, this is also a system. It also needs to be regulated, controlled. So for that purpose, also we have humoral system to control, we have neural control also. Neural control is done by, nervous control is done by, GI tract is unique. It is having its own nervous system. It's having its own nervous system. We call it enteric nervous system. Similarly, GI tract is also having its own humoral mechanism, means endocrine system. We call them controlled by GI hormones, GI endocrine system. Neural control, as I said, basically done by enteric nervous system, ENS. They regulate the functioning of the whole GI tract, their movement in particular, their secretions in particular. Though they can control themselves, their functioning of GI tract, they have also control from CNS, central nervous system. Central nervous system has sympathetic and parasympathetic autonomic nervous system. So both these systems, sympathetic and parasympathetic nervous system, has control on the enteric nervous system. We learn that after 1 week. So this is about neural control. And for humoral control of GI tract, there are hormones. The hormones are actually transmit signals to its target organs, to its target cells. So this transmission of signals, cell-to-cell signals, are done by the hormones. We classify them, the mechanism by which they transfer the signal. We classify them into three. One is hormonal, endocrine. Another is paracrine. Signaling, paracrine signaling, hormone signaling, endocrine signaling rather, and autocrine signaling. So, by these three mechanisms, the hormone sends signal to its target cells. What are these three? Endocrine, paracrine, and autocrine signaling. Autocrine signaling means the cell will produce one ligand. You know what is ligand? Hormone. The cells will produce one ligand. After their liberation, secretion, they will bind with a receptor present in the same cell, on the surface of the cell. And binding of this ligand with the receptors will control the functioning of itself. Means it is a self-controlling process. Autocrines are self-controlling. The same cell will be liberating ligands. This ligand will be binding with the receptors of the same cell. There are receptors for the same ligand. Now, this ligand will produce action within this cell. So this type of regulation is called autocrine regulation. Example of this is interleukin 1. It is secreted, is one of the, this is secreted from this cell, and it has got receptor of this cell, then again it binds with this same cell, and it will produce regulation of the cell. Then the paracrine, the cells, the secretory cells and the target cell, they are lying side by side. Para means side. So adjacent to the cell, you see, after secreting from these cells, they will bind with the neighboring cells. There are receptors and will produce this action. This type of regulation is called paracrine regulation. We'll find there are some GI hormones which acts like paracrine. But for endocrine, what happens? Suppose he is secreting hormone. Now this hormone, this cell is secreting hormone. Now this hormone will be liberated into the extracellular fluid. From there it will enter into the blood vessel. And through blood it will reach to his target organs. So there must be blood who will be circulating them. But here in case of paracrine we saw, there is no need of blood. Just tissue fluid. In between there is tissue fluid, little tissue fluid. So from this cell to cell, no need of transferring by the blood. And autocrine, very shortest route. After liberating from the cell, again it is binding with the cell and controlling its function. So GI hormones, They are endocrine as well as paracrine. And sometimes we will find the autocrine also. The cells that produce hormones in the GI tract, we call them enteroendocrine cell. What is the term we use? Enteroendocrine cells. Now these enteroendocrine cells, they do not make any gland within the GI tract. We can see thyroid gland. So here the hormones are produced in a gland. Pituitary gland. So hormones are produced in a gland. In suprarenal gland, the corticoid hormones, glucocorticoid, mineralocorticoid, sex corticoid, adrenaline. So these hormones are produced in a gland. But no such gland is present in the GI tract who will be liberating the GI hormones. Clear? There is no specific glands. These hormones are, the cells are actually, they are producing it. And these cells are present throughout the GI tract. You will find it, these enteroendocrine cells in stomach, in intestine, in large intestine, there you will get everywhere they are present. These hormones we classify into different categories depending on their composition, how they are made, the chemical characteristics, and their function. So one group is gastrin family. Gastrin family. Another group is secretin families. There is somatostatin family. There is P substance family. So these are the main families. Means under these families there are different groups, different GI hormones. So we will not be studying all these, only the important ones we will be studying. Because a lot of varieties are there. A lot of somatostatins are there, a lot of GIPs are there, so they are different. So we will only go through the important ones. Among the gastrin family, there are two main hormones, GI hormones. One is cholecystokinin, CCK. It is also named as CCK-PZ, pancreazymin. Cholecystokinin, CCK, cholecystokinin. Contraction. Kinin, contraction. Chole related to gallbladder. So this hormone basically will cause contraction of the gallbladder. They will be secreted from the intestine. They must, to reach the gallbladder, they must come through blood. So it is not a paracrine. It is nor a autocrine. So another group, another member of this is gastrin. These gastrins are secreted from the stomach, acts on stomach. Again it has to pass through the blood, then again come to the, because the target cells are not here, it will be apart. Its target cell may be in intestine. So they have to pass through blood. Similarly, secretin is there, secretin group. So under this group there are five important hormones. We study these hormones. So let us have some overview, summarized overview of these hormones. Main hormones. What are their stimulus? How they get stimulated? These cells has to be, they are not producing automatically. Somebody has to stimulate. So what are these stimuli? And who are the producer? Who are the cells that produce this thing? And what is their action? Means their main functions. Okay, let's see. Our syllabus says you are to learn, you are to list, enumerate, means listing different GI hormones. You have to discuss their functions and discuss their regulations. These are the main aspects what you have to know from the GI hormones. As I already told, as you know, aware, what are the main functions of the stomach? Why can you tell me 5 important functions of the stomach? Ingestion of food. Not stomach, GI tract. Ingestion of food. Then this food has to be motility. Transferring this food along the GI tract. Means GI tract movement. Okay? So this is done by the muscular coat. And to control this movement, there are nervous system. And this nervous system are sometimes affected by this hormonal system. So they lie in between the muscular coat. There are longitudinal muscles, there are circulatory muscles. So in between, throughout the GI tract, in between lies these neurons of the of this nervous system. We'll discuss in detail how they are constructed. And another important function of GI tract is digestion and absorption. And the undigested part, unused part, has to be eliminated. So that process is also done by the GI tract. And another part of it is to facilitate the absorption and digestion. They secrete, their accessory organs secrete some fluids containing different enzymes needed for digestion, needed for neutralization function and other functions. Okay, so beside this, they also have endocrine function. They are producing this. So these are the main functions of the GI tract and its accessory areas. Say for example, only in the stomach, these are the gastric glands of stomach. Here you will find G cells that will be secreting gastrin. You will find D cells they will be secreting somatostatin. So we know from where glucagon is secreted. Glucagon. From where insulin is secreted? Pancreas. Okay, but they can also be secreted from the GI tract, from the cells of the GI tract. Some insulin-like action is like insulin. Some insulin-like substances are also secreted, and their function is also, in addition to functioning in GI tract, they can also cause secretion of insulin. They can also function as insulin. Dual function. Okay. But their role is not like the role of human cells. Then if we have diabetes, means insulin is produced by the beta cells, then this insulin can do the thing for us. But this is not sufficient. This is sufficient for maintaining the GI tract function. What role it is performing, that role can be performed with that much concentration. So as I said, I will be giving you some overview. I have shared this file in your group. I sent it to, I think everybody got it. So while you will be going out of this class, go through it. You will be learning in details from there. So here you see, first hormone is gastrin. And what are the stimuli for this? Protein and its digested products. In presence of protein in stomach, it is secreted from stomach. Gastrin is secreted from stomach. So whenever you will taking food, this will be containing protein. So this will be coming in touch of G cells, gastrin producing cells. Immediately there will be, the cell will secrete. They are having lot of zymogen granules. So these are packed with gastrin, this protein. So when it touches or it finds the presence of protein, immediately it will secrete pepsinogen. It will secrete hydrochloric acid. Okay, mainly pepsinogen. This will cause secretion of, there will be secretion of gastrin, and it will cause, what it will cause? Gastric acid secretion. Okay, so this gastric acid secretion is done by two cells mainly. One is peptic cell, another is parietal cells. Peptic cell means chief cells. So they are having in their basolateral surface some receptors, and these receptors are for gastrin. So gastrin is secreted in presence of protein. Now this gastrin will bind to the secretory cells. That is, what? What are these cells? Parietal cell and parietal cells will be secreting HCl. Is it sufficient? There should be secretion of enzyme. So the same cell is not secreting enzyme. So there are other cells, chief cells, also called peptic cell. So they, both of them are having receptors. So now the gastrin will bind with these receptors. When it binds with the peptic cell, they will secrete pepsin. And when they will bind with the parietal cell, they will secrete hydrochloric acid and intrinsic factor. So that way, gastrin is stimulated and produces effects. You could follow this? Okay. Then comes the secretin. It is secreted from intestine, not from stomach. It is secreted from, what are the cells? S cells. Secretin starts with letter S, so it is the S cells. So S cells of intestine, these cells are actually present on the brass border epithelium, ekdom close to intestinal lumen. So when food bolus kind coming from the stomach entering into the intestine, so that will be having hydrochloric acid. So now the food bolus along with hydrochloric will come in touch with the brass border epithelium. So there are the cells, S cells. Now these S cells will be stimulated and it will produce secretin. Now the secretin will act on, their function is pancreatic and biliary bicarbonate secretion. Biliary secretion and bicarbonate secretion mainly. Alkaline secretion. So we know in pancreas, there are duct cells, echinal cells. These echinal cells are mainly responsible for enzyme secretion, pancreatic enzyme secretion, and when the fluid along with this enzyme secretion will be passing through the ducts, these ductal cells actually secrete alkaline substances, bicarbonate ions. So secretin stimulates those cells, ductal cells of pancreas to release more and more alkaline material, that is bicarbonate material, so that they were stimulated by acid. They are alarming, it is lot of acid. So neutralize it. Otherwise, how our enzymes will act? We need the alkaline medium. So immediately its secretion has to be increased. Bicarbonate secretion has to be increased. So these bicarbonate ions are mainly present in pancreas, also in gallbladder. So they are storing bile. Bile is having lot of bicarbonate ions. Means these are alkaline. Pancreatic juice also alkaline. So who are secreting this alkaline juice? Ductal cells. Cells around the ducts. Intercalated duct, pancreatic duct, their wall is secreting this bicarbonate ion. Now the juice along with the bicarbonate ions and the juice from the bile, through bile, will be entering into the duodenum and they will neutralize the acids coming from stomach. Okay, so that is not the main, that is done. Okay, neutralization is done. Then digestion has to be done. So for that purpose, the pancreatic cells, echinal cells, they are also secreting enzymes. They are mainly, so with that enzymes, the rest part will be done. That is, digestion will be done. Gastric inhibitory peptide, it has two names. When it was found that gastric inhibitory peptide is also able to secrete insulin, they can also stimulate the pancreas. So then its name, it has been given another name. What is the name? Insulin. Not insulin. What is the name? Another insulinotropic hormone. Okay, we are coming into this. So insulin release, it is produced by the K cells, and it is, everything. GI, gastric inhibitory peptide, it is stimulated by everything. Means food bolus, when entering into the duodenum, it will distend the wall. This is one stimuli. This is one stimulus. And its acidic content, this is another stimuli. And partly digested proteins are also there. This is also stimulus for stimulating gastric inhibitory peptide. So when they are secreted from the K cells, they will cause insulin release. So to release insulin from pancreas, they must be absorbed into the blood. Through blood, they must be circulated to the pancreas. There it will cause secretion of insulin. And since the product is acidic, so food has entered from stomach into the duodenum, into intestine. So there is no need of further secretion of hydrochloric acid. So it will cause inhibition of secretion of hydrochloric acid by the stomach, because its function is already over. Now if you secrete hydrochloric acid more, then it will be the action will be problematic, because this acid will be entering into the duodenum and they will change the environment acidic. So enzymes will be unable to act. So that's why it will inhibit secretion of hydrochloric acid. So main function, inhibition of hydrochloric acid secretion. When food will be entering from stomach into the diet, then only. In presence of this will cause inhibition of hydrochloric acid secretion. At the same time, secretion of insulin from the pancreas. Why? Get ready. A lot of glucose is coming. So if you are not ready, then there will be shooting of blood levels of glucose, and this is not wanted. So that's why beforehand they get it is so that it is prepared. It can control the sugar level. Whenever we eat, lot of glucose will be formed, so they will be absorbed. Okay. So absorbed into the blood level, so it will rise. So what happens actually? Insulin doesn't allow this. Immediately insulin secretion is made and they let this glucose enter into the cells. So blood levels comes to normal level. When blood glucose level comes down below normal, then what happens? Glucagon is secreted. And it will raise blood sugar level. How? There is some mechanism. Some stored glycogen we are having. Insulin has made it. Okay. So now this glycogen will be broken down into glucose and then will be released into the blood, so that blood glucose level comes to normal. There is no hypoglycemic shock. Okay, motilin. Very important hormone, GI tract hormone, motilin. The name itself says it is concerned. Motilin concerned with movement, particularly of GI tract. When in between the meals, lunch, then you will be taking dinner. 2 main meals. So in between, what has to be done? The GI tract has to be cleaned. With this movement, we clean the GI tract. When you go to a hotel, okay, and when you leave the hotel, check out, then housekeeper comes, enters into the room. What they do? They clean everything. New bed sheet, everything is, floor is cleaned, bathroom, everything is cleaned. New customer will be coming, so that he can get everything ready. This gentleman, motilin, also prepare in between meals, because another meal will be coming. So before that, I should clean everything. There should not be any particles left behind. Should not be any enzymes or like that, any acids. So evacuate them. So with this movement, motilin makes this movement. That's why we call it a housekeeper of GI tract. Interdigestive enteric complexes. Interdigestive means inter meals. So we are actually getting ready for the digestion of the next meals. So for that purpose, to make it ready, there is a movement of GI tract is needed. Migrating motor complex we will be learning, very important. See, when you take food, usually it happens. It also happened to you also. But by training or habits, you have changed it. What is that? After feeding, full stomach, and after few minutes, then you feel like going to toilet for defecation. Why it is there? When we have taken the food, then ultimately this food will come to the large intestine and lot of stool will be formed. So before, if you have already some stools, pass it. Because they will be coming, they will be forming stool, and this stool has to be given space. So before that, you eliminate it. So we call it migratory motor complexes. We learn it, how this has been done. So motilin will increase GI tract motility. Okay? And these are other hormones. What are these? VIP, glucagon. As I said, glucagon is secreted from alpha cells of pancreas. But these are also secreted from, where is glucagon? Alpha cells they are also A cells in stomach, L cell in intestine. So in addition to cells of alpha cells of pancreas, they are also secreted from A cells of stomach and L cells of intestine. What is their purpose? Hyperglycemia. Obviously, these are insulin and glucagon, they are opposing in action. Whenever you take food, digested carbohydrate and form lot of glucose, this glucose will be entering to the blood sugar will be raised. So we cannot allow this raised blood sugar, they have to enter. Why we have taken glucose? For energy, for the purpose of the cells. So this will not do anything. If it only remains in blood, this has to pass to the cells. But without the help of insulin, it cannot enter into the cells. So that's why a lot of insulin is secreted. So now this insulin will cause shifting of glucose from blood into the cells. So as a result, what will happen? Blood sugar will be lower. So that's why we call the insulin has hypoglycemic action. It reduces blood sugar level by absorbing it into the cells. It reduces blood sugar level. But the glucagon is having other effect, opposite effect. When blood sugar level fall below the normal level, immediately these cells are stimulated because blood sugar level has to be maintained. So what it does, it stimulates liver to stimulate the process of glycogenolysis, to stimulate the process of gluconeogenesis. Okay, and also it inhibits liver enzyme so that it doesn't cause any glycolysis. There will be breakdown of glucose. So it will prevent glycolysis. It will increase glycogenolysis so that more glucose is formed from stored glycogen, and gluconeogenesis from non-carbohydrate substances, glucose has to be formed. Now this glucose will be entering to the cell and reach the blood sugar level. Now this sugar can be utilized by the peripheral cells. You are fasting, you are not taking any food, what to do? You need energy. Then glycogen will come into, not glycogen, glucagon will come into action, and it will increase the blood sugar level so that this sugar can be supplied to the different cells. So glucagon will cause hyperglycemia. Its main purpose is to increase the blood sugar level. So if this action is called increasing blood sugar level, means hyperglycemia, blood levels of glucose will be increased. So this condition is called hyperglycemia. Next, substance P. It is secreted from anywhere of the body. Nowadays, a lot of researches are going on on these P substances. So in our GI system, it increases intestinal motility. This is not the only function of substance P. But since we are concerned with the GI tract, we will be knowing only this. Okay, this is the main function. And these are secreted from nerve endings mainly. Now, when these nerves are stimulated, their ending will produce P substances. Like acetylcholine, adrenaline, histamine, these are the neurosecretory substances. P substance is nothing but neurosecretaries. Neurosecretions. Defenses, we have learned about this before. It generates appetite. It causes, that's why we call it hunger. When we feel hunger because of ghrelin. What is this? Defensin. Its action is immunity. See, a lot of bacteria is there in the intestinal flora. A lot of bacteria. Some are very beneficial and some are not harmful. So it balances between harmful and helpful bacteria, which are to keep and where to act against this necessity. There will be necessity of killing the exogenous bacteria. So they are stimulated by, see, what are the stimulating factor? Defensin, live or dead bacteria. So in presence of them, there will be inflammation, and by doing this, it will be destroying those. If it is not at all harmful, if it is good for us, so it will allow to survive those bacteria in the intestinal flora. Somatostatin, these are also having multiple actions. They are also secreted from pancreas. They can also be secreted from stomach, small intestine, and these cells of islets of Langerhans. In islets of Langerhans, IL means islets of Langerhans. These cells means delta cells. We know in islets of Langerhans there are alpha cells. Alpha cells is responsible for glucagon release, and beta cell is responsible for glucose release, and this is responsible for somatostatin. So what are these action of somatostatin? It inhibits gastric secretion, It inhibits vasoactive intestinal peptide. It inhibits GIP, gastrin inhibitory peptide, and also inhibit motilin. So these are the functions of somatostatin. They also regulate the secretions of, these are actually inhibitory. Secretions of insulin, also they inhibit it. Okay, when we will be learning about endocrine of pancreas, means functioning of insulin, glucagon, then we will find what is the role somatostatin is performing there. Another important thing is also secreted from pancreas, that is somatomedin. Somatomedin is also very helpful for growth. So these are the overview. So you try to learn this basic thing. If you learn this basic thing, this chart, now you will be able to answer any question related to GI hormones. So you go through it. And these, here we will be studying in detail, though it is not required too much, this much. It is actually required, very much required in PG level. However, nature of peptide, it is a peptide hormone having some number of amino acids, and it is secreted by G cells of antrum, not of pylorus region, antrum region. So from there it is secreted, and it binds, G cell binds with cholecystokinin B receptors to stimulate the release of histamine in enterochromaffin-like cells. It binds to cholecystokinin B receptors to stimulate the release of histamines, and as I already read, it induces the insertion of proton pump. What is this? Proton pump. So thereby it will activate. When the activity of proton pump is increased, what will happen? A lot of hydrogen ion will be released into the gastric juice. So means hydrogen ion, at the same time there will be chloride ion liberation. So they will combine, will form hydrochloric acid. Now this will increase the hydrochloric acid level in the stomach, and it is needed for various purposes. One is for activation of pepsinogen into pepsin, and other function to kill the bacteria, foreign organisms, and to make the environment there acidic. Whose points need to be clarified to you? Tell me. Tell me the number. Say 7, 8, 5, 4, 3. Which one? All points are clear to you? Needs clarification? Explanation? No. I already mentioned about this number 8 points. Contraction of muscles at the gastroesophageal junction. What is there at this junction? If the muscles are contracted, what benefits I will get? Stomach is in peristaltic movement. So it is crushing the food. So by pressure, because of pressure, the content may enter into the esophagus. So it may lead to reflux. It may cause reflux esophagitis, inflammation of the esophagus. It may cause vomiting. To prevent that, so those muscles are constricted so that there is no reflux. So this is another function of mechanism of action. Go through it. Release of gastrin, binding with receptors of parietal cells. There will be formation of IP3 as second messenger. Receptor, this is the membrane. This is the receptor bound. Gastrin became bound to it. So the other end of this protein inside the cell, that will produce a second messenger. And what is the second messenger here? IP3. Now this IP3 will cause release of calcium ion. Now what this calcium ion will cause? Activation of protein kinase. When this protein kinase, this is within the cell of the parietal cells. So activation of protein kinase will cause activation of proton pump. So proton pump at the apical surface, GI lumen and the parietal cells, this is the apical membrane, here are the proton pump. So this pump from bicarbonate ion H2CO3 will be dissociated into H ion plus bicarbonate ion. This bicarbonate will be going to the blood and hydrogen ion will be pumped into the lumen. And from the cell, from blood, chloride will be entering into the parietal cells, from parietal cells that chloride will be coming into the lumen. So we are getting hydrogen ion and chloride ion, both of them will be combining and will form hydrochloric acid. So this is the action of the proton pump. Its activity is increased, there will be more formation of hydrochloric acid. Okay, in this way, gastrin it stimulates the parietal cells and helps in releasing hydrochloric acid. Regulation. So these gastric cells, these G cells, they are stimulated by these factors. Distension of stomach, G cells will be stimulated, they will form gastrin. Vagus stimulation. Vagus stimulation, we already told repeatedly, vagus nerve has got a inhibitory effect on GI tract. But you see here, what is there? Vagus nerve coming from a 10th cranial nerve and it doesn't pass through spinal cord. It's a cranial nerve, no? It's coming out from the cranial. Then it will coming to the organ where will be supplying. Okay. So in the target organ there will be a ganglia. So this nerve will be ending in the ganglia. So this nerve is a pre-ganglionic neuron and from the ganglia post-ganglionic neuron will be formed and this post-ganglionic neuron, they will synapse or they will end at the base of the parietal cells. And when this nerve is stimulated, so from their nerve endings, acetylcholine will be liberated and they will cause secretion of hydrochloric acid. Okay, so this is, actually this post-ganglionic nerve are two types. One will produce acetylcholine at the nerve endings and another part will produce GRP, gastrin releasing factor. Now this GRP, gastrin releasing factor, has got receptor at the base of the parietal cells. So now GRP will bind with these receptors and make necessary change in the parietal cells, means secretion of hydrochloric acid. So products of protein digestion also positive. It causes secretion of pepsin, it will digest protein. So there will be formation of protein products like amino acids, dipeptides. So they will again cause secretion. This is called, they will have further secretion of pepsin. So this is called positive feedback. But we have two types of feedback mechanism. One is positive feedback, another is negative feedback. Okay, we have seen the positive feedback mechanism in case of trypsinogen action. Okay, so trypsinogen is released, it is activated by hydrochloric acid. Pepsinogen. For trypsinogen it is enterokinase. So some enterokinase converted trypsinogen into trypsin. Now the trypsin itself will further stimulate inactive trypsinogen. Continuously will do it. Positive feedback. And negative feedback is Suppose one product is formed by the action one product. When this product formation is increased, they will inhibit the formation of this product. But too much is this not good. Okay, so this type of negative and positive feedback mechanisms are there. And inhibited by, so you have, these are the regulation, very simple. If you go through, you will be understanding. Very simple one. So you go, these are the regulations, how this regulation is made. Cholecystokinin, main GI hormones, you study the main GI hormones.